The TARMAC study was an investigator-initiated Phase II study that looked at the combination of the BTK inhibitor Ibrutinib and the 4-1BB co-stimulated CD19 CAR T-cell Tisagenlecleucel in mantle cell lymphoma. The rationale for the study is that patients with relapsed refractory mantle cell lymphoma still remain in need of improved therapies. Outcomes after first relapse are modest, at between 18 and 24 months of progression-free survival, and this progressively shortens with each subsequent treatment line. There are a number of active therapies in relapsed refractory mantle cell lymphoma, and the most commonly utilized worldwide would be BTK inhibitors. But these remain imperfect in treatment of relapsed refractory mantle cell lymphoma. They don't lead to deep and durable responses, and most patients that are exposed to these agents eventually progress on them. We're lucky to live in an era of a lot of effective new and emerging therapies, particularly immunotherapies and T-cell redirecting therapies in particular. And these include bispecific antibodies and chimeric antigen receptor T-cells. But one of the problems is that patients that are progressing on a BTK inhibitor may not actually ever reach a next line of treatment, just because of the nature of mantle cell lymphoma and the aggressiveness of some relapses. So we sought to test the combination of BTK inhibitors and CAR T-cells, both in second and in subsequent lines of treatment. One of the key rationales behind it was that there is potential synergy between the two agents. Ibrutinib has direct effects on mantle cell lymphoma through inhibition of BTK, but also inhibition of other enzymes that are important for both B and T cell development. And so one of the rationales that has been demonstrated in previous CLL studies by a number of groups is that Ibrutinib can affect and perhaps improve the CAR T-cell product. So we enrolled 20 patients in our study at 3 centres across Australia. They were all treated with Ibrutinib for at least a week before the CAR T-cells were collected for manufacture. Patients were then able to receive additional bridging therapy, which allowed patients with rapidly progressing disease to be considered for the study, as they had that option to bridge before receiving the CAR T-cells. Patients then received the CAR T-cell product and continued Ibrutinib throughout this phase, and then for a period of 6 months after treatment, at which time the Ibrutinib was ceased in patients who were measurable residual disease negative. We really wanted to maximize the benefit from the Ibrutinib around the CAR T-cells, but also deliver on the promise of CAR T-cell therapy, which is a one-off, time-limited therapy. And so where we felt able to, we really wanted to stop ongoing therapy and hope that patients remained in remission on no active treatment. 20 patients were infused with CAR T-cells, and the overall response rate was 80% at our primary endpoint, which is 4 months after infusion. This compares pretty favorably with other treatments in this area, and we sought to look into other factors, including what happened to people who were BTK naive versus those that had previously had a BTK inhibitor, and both groups did well on the treatment. We also sought to really characterize the lymphoma itself to understand, because one of the key unmet areas of need in mantle cell lymphoma is in high-risk molecular mutations such as TP53 mutations, which perform poorly with chemotherapy and a lot of the novel targeted agents. We found that the treatment combination was highly effective in this group. So we had 8 of 20 patients with aberration of TP53, and they performed just as well as patients without these high-risk molecular mutations. We've followed patients for 13 months now as a median, and 75% were free of progression at the 12-month mark. So at this early stage, this is a highly effective treatment combination in patients with or without prior BTK exposure, with or without TP53 mutation, and at this stage appears to be a durable treatment option for them.